Stroke is a devastating medical emergency that 1 in 4 of us will experience during our lifetime. It is caused by a lack of blood flow in a particular area of the brain. Your blood supplies oxygen and nutrients to your tissue, and without this, brain cells begin to die within minutes. Over the course of the minutes, hours, and even days following a stroke, this damage continues to spread. The current treatment for stroke is to return blood flow as quickly as possible, either by clearing a clot or by repairing a broken blood vessel, but there is no treatment to reverse the damage that has already been done. For my research, I'm developing a way to test potential stroke therapies in a dish. In the Hoffman Kim lab, we have developed a 3D brain microtissue model, colloquially known as a mini brain. These mini brains are tiny, about the width of a tip of a mechanical pencil, barely visible to the naked eye, and yet they replicate many of the aspects of an actual brain. They have the same cell types as a brain, the neurons communicate with each other as they would in the brain, and they have many of the same physical properties, including tissue stiffness and cell density. We can also make and maintain thousands of them at a time, making them much more cost-effective than the traditional animal model. All of these characteristics indicate that the mini brain would be a good model to screen potential therapies. As a neuroscientist, I'm using these mini brains to observe what occurs after a stroke-like injury. To induce this injury, I deprive the mini brains of oxygen and glucose, simulating that lack of blood flow. To do this, I replace the media that the cells use to grow with glucose-free media, and I place the mini brains in an anaerobic chamber. This creates a low oxygen, low glucose environment, similar to what your brain experiences during a stroke. After this injury has been induced, I prepare the mini brains for one of several outputs. An example of this is that I compare the morphology or the cell structure between a healthy control and an injured mini brain. As you can see above, in red are the neurons. In the control, we can see that they have a nice strand-like structure. This allows the neurons to communicate with one another. But after a stroke-like injury, this structure is disrupted, and so is the communication between cells. In green, we see another cell type, an astrocyte. In the control, they have a healthy morphology, but after the injury, they get a bit larger and bushier, and this is a sign of brain inflammation. My model replicates many of the injury pathways that we see in stroke, including that disruption in neuron communication and signs of brain inflammation that I've shown some evidence for, as well as a general decrease in cell health. The next step in this type of research is to test a potential therapy. And recently, we've shown that N-acetylcysteine, an antioxidant drug, is able to prevent some of the effects of a stroke-like injury. As you can see in red, again, we see the neurons. As before, in the injured mini brain, we can see that the strand-like structure is disrupted. But if we pre-treat with N-acetylcysteine, this structure is preserved, and so is this communication between cells. While this is not a perfect solution to stroke, it does show the feasibility of using our model to screen potential therapies. My hope is that we can use the mini brains to get drugs from the lab into patients in a more cost-effective and efficient manner and help mitigate the devastating effects of stroke. Thank you.